welcome friends and partners of Melbourne Connect, both here in the forum and those joining us live and online through the live stream. I'm delighted to welcome you to the Melbourne Connect Oration, a centrepiece of the Melbourne Connect Innovation Week Festival. My name is Georgia von Gutner, and I'm the Director of Innovation Precincts for the University of Melbourne, and I'll be your MC for this evening. I would like to begin by acknowledging the traditional owners of the land on which we meet, the Wurundjeri Woi-wurrung peoples. I pay my respect to their elders past, present and emerging, and I extend that respect to any First Nations people joining us here or online tonight. Responding to the grand challenges all around us, our goal at Melbourne Connect is to drive the development of new technologies, ways of working, and solutions that are digitally enabled, data-driven, and socially responsible. In just 2 short years, we've become an important part of the Australian innovation landscape. Melbourne Connect is already fostering impactful connections between our industry partners, government and the social sector, with the university's interdisciplinary expertise and our student talent. We're also inspiring and training inventors and entrepreneurs and connecting them with investors and wider mentor communities, as well as facilitating school age and wider community involvement through the exhibitions and programs of Science Gallery Melbourne and the other programming that takes place here at Melbourne Connect. And very much on theme, tonight's oration topic is unlocking mind power. And it's all about the development of a brain-computer interface for people with paralysis. The oration will be delivered by Professor Prof Opie, a biomedical engineer and world expert in neural interfaces. Nick will be introduced by our Acting Deputy Vice-Chancellor Research, Professor Mark Hargraves, and following the oration, we'll have time for your questions, which we very warmly welcome. Can you please now put your phones to silent? And I'd like to share a clip from Professor Edward Hovey, who's the Executive Director of Melbourne Connect and himself an eminent computer scientist. Ed is currently overseas, responding to and attending to a commitment he made many years ago. He would have loved to have joined us. Let's hear from Ed.
Welcome to the oration of Melbourne Connect's Innovation Week. My name is Edward Hovey. I'm the Executive Director of Melbourne Connect. It's my pleasure to welcome you here this evening. I'd like to acknowledge first the Wurundjeri people of the Kulin Nation, our nation's first scientists and engineers, and their elders past, present and emerging. We've been working hard at Melbourne Connect to make this a focal point of interesting innovation and academic discussion. Today you're going to hear from Professor Prof Opie, who's going to talk about the brain and what you can do with the brain. And believe me, this is only the beginning. We're going to hear a lot about the brain in the future. When the brain starts meeting AI, you ain't heard nothing yet. There's a lot more coming. Thank you.
And with that, it is my absolute privilege and pleasure to introduce our speaker tonight is Professor Mark Hargraves, AM. Professor Hargraves is the university's acting Deputy Vice-Chancellor Research, which means he has responsibility for the research strategy of the university and facilitating major research opportunities in collaboration with our faculty deans. Mark is a Professor of Physiology and with focus on the physiological and metabolic response to acute and chronic exercise. He's a Board Director of the Florey, a leading centre of brain research, and has led the university's research collaboration and partnerships portfolio for many years, which includes development of the university's innovation precincts, including Melbourne Connect. Please welcome Professor Mark Hargraves.
Thanks very much, Georgia. And again, on behalf of the university, welcome to this evening's oration. And I too would like to pay my respects to the elders, past and present of the Wurundjeri, the original innovators, if you like, on these lands. It's a great pleasure to be acting DVC at the University of Melbourne, and every day I get to see the extraordinary activities of our researchers and the support that they receive from professional colleagues who support various parts of the research journey. And our strategy over the years has really been focused on excellence and impact. And I think for many years, the university researchers have had that commitment and in various ways have had impact on the communities that we serve. In recent years, we've sought to be more purposeful in providing the opportunities for research translation for greater impact. We recognise that much of the work we do is investigator-driven, with a commitment to the scholarly traditions of the disciplines in which they work. But while that's necessary, it's often not sufficient to really take those learnings into practice. And so we have a commitment to impact in the scholarly and creative area, but increasingly we're looking to have an effect on policy, on economy, on health and well-being, and there are many examples across the academy where this can be demonstrated. Some of the initiatives we've undertaken is to bring people together in precincts such as the Melbourne Connect building and precinct, bringing government, industry, innovators, our researchers together to try and catalyse the interactions that will lead to more impactful outcomes. Another initiative is to foster interdisciplinary activities, and we absolutely committed to the strength of our disciplines, but bringing them together, they are the foundations of opportunities to work together. And I think tonight you'll see a wonderful example of interdisciplinary work. I remember the first paper in Nature Biotechnology with engineers, material scientists, clinicians, neuroscientists, all working together to validate a device, and the subsequent work to trial it in humans and to take it to the clinic to help patients. So in that vein, it's my pleasure to introduce Professor Nick Opie, who, as Georgia said, is a biomedical engineer, an expert on neural interfaces, and also the CTO of Synchron, which is a company that has taken Stentrode internationally and with the intent to translate great research from our scientists at the University of Melbourne into tangible benefits, improving the lives of people where this device can really make a difference. So please join me in welcoming Professor Nick Opie to deliver this year's Melbourne Connect Oration.
Yeah, thank you. Yeah, it's a pleasure to be here. Certainly, the journey that I've been on has been a long one and a challenging one, but a really exciting one as well. And so I'm the professor of the University of Melbourne, running the Vascular Bionics Lab out there, and at the same time, sort of working as the founding director of Synchron. And I was just speaking to Georgia before, and I think it's really critical, certainly in retrospect, when you have those two things going together at the same time. I'll get into this more as I go through the slides, but there comes a point where you're required to do some sort of tests that don't really have a lot of academic value, but you need to do them for FDA approval. And there comes a time where you need to raise capital that goes beyond what the grants can afford. And for that reason, it's really important to have a company, but you need to have the university and some sort of research institute to help you do the fundamental knowledge and get the ball rolling, as I will do now. So we're developing brain-computer interfaces, and for us, the perspective has changed a little bit over the years, as it needs to. Where it started off where we wanted to make robotic arms and exoskeletons and things for people who couldn't move. What became really valuable and important as we went along the journey was you really need to speak to the clients and the patients, or whoever it is that you're making the technology for. And it was really over that journey that we realised that the thing that a lot of people, particularly with motor neurone disease, were after was how to communicate and how to do that with a computer. And certainly over the last sort of decade or so, any of you kids will understand this well, that handwriting letters and posting them isn't done anymore. Everything's done online. Everything is done through messages. Some people in the crowd, and I'm not going to judge you, probably have Facebook profiles and Twitter accounts and all that sort of stuff and tell each other about what you had for breakfast, but everything's done online. And to get online, you need to use your hands. And so really for what we came to think about is how can we make a technology that will allow people to communicate, which isn't just with your voice anymore. And I think a great example of this is this guy, Stephen Hawking, an incredible individual, and one who has motor neuron disease, and in this disease, the brain is fine. Like, he's a perfect example of this, writing some incredible books, an amazing individual, and it was his body that was sort of starting to let him down. It was the ability of his brain to connect to his body that stopped working and prevented him from walking or typing or from doing sort of conventional activities. And certainly the number I've got here, the 100 million people that can't access the digital world, and I think that's where it's important, because the world is becoming one where your fingers is what's doing your communication. And for a lot of people on one end of the spectrum, you've got paralysis, spinal cord injury, stroke, where you're very disabled in terms of your physical ability, all the way through to people who can't move their hands because they've got arthritis so bad that they can't type. So these are all people that in the future will benefit from some technology that allows them to reconnect. Sciencey slides. I'll go through these quickly. I've got some really nice videos at the end, so I'm going to make sure I don't spend too much time talking about this sort of stuff. But essentially, the way your body works is your brain comes up with some command. If you want to move your hand, for example, there's a part of your brain in the motor cortex that becomes active. And when that becomes active, it sends a message through to, in this example, your hand and your finger, and you can use that to use a phone. There are a huge number of reasons why that might not work. Some are, genetic, some are from damage or trauma or accident, and largely the end result is the same. Your brain thinks about a signal, the signal's generated, but it just doesn't go anywhere. And there's many ways along this pathway where damage or disease can stop this signal from progressing. And so what a brain-machine interface does in general terms is it bypasses all of this stuff. And you go straight from the brain and you plug it directly into a phone or a computer or some piece of digital technology. And in this regard, it doesn't really matter what caused the problem or what disease or what etiology or any of that. If you have a brain that works and you want to communicate with something, then you can do that. And that's the benefit of this technology. And certainly, there's a lot of discussions we've had with the FDA and other regulatory bodies who are used to having a problem to fix a particular condition or disease. And for us, we're saying we don't care. Like, we want to fix all of it. If the problem is you can't type, that's the problem, not because you have a spinal cord injury or because you have had a stroke or because you're missing fingers or whatever it is. So that's an ongoing challenge, and I think it's something that they're very receptive to, which is great. As I mentioned, different parts of the body can do different activities. It's all part of the motor cortex, and I suppose we'll get into that a bit later. But essentially, just flag this in your mind that there are different parts that do different things, and that's how we can do different stuff. I'll get back to this. So invasive BCIs. Let's start here because they're the oldest. Invasive BCIs or invasive brain-computer interfaces are ones that conventionally will require getting inside the head. So I suppose there's non-invasives, which are, I sort of call them toys, but they're ones that go on the skull or the scalp. They don't require any surgery, which is great, but they also don't get the signal that you need because the skull blocks it. The skull's very good at blocking the signal that's coming from the brain. So I sort of put them to the side. They're regulated differently and so forth. But in terms of the invasive BCIs, or the ones that go into the brain to get the signals, the way they used to do that, and don't worry, I don't have any gruesome photos, I don't think. But the way you used to do that is get a saw and cut off some of your skull, and then get some of these needle electrodes and shoot them into your brain. And it's all very barbaric, but it's shown that it works really well, right? There are people like this gentleman here who he's had to have his removed recently, but at least for 7 years or so, he had this device implanted in his head, and he was able to do some amazing things with it. He could play Guitar Hero. He could, he could use a computer. He could communicate again, but he couldn't do it outside the lab. He could, if he wanted to use this, he had to go into this lab and get hooked up to all this kit, and he had to, really give up the remaining bit of his freedom and independence to try and get some sort of restoration of ability. As you can see from this lady, though, I mean, it works quite well. So she's got these devices that are implanted into her brain directly. They're recording the signals when she's trying to move left or right or up or down, and using this robotic arm, and this is many years old, but it's one of the earlier ones. She's able to feed herself and control this arm to in a way that she hasn't been able to do before. So some of this technology I think is just absolutely incredible. Again, can't be used at home. Here's another example of a lady who's typing much faster than, I used to say my dad, but I think he's getting better, so it's probably faster than I can type now. But she's using this device, she's locked in, she can't move her face, she can't move her hands, can't move her body. But with this device that's implanted in her head, she can move around a cursor on a screen and start typing messages. You can converse with loved ones and so forth. And some of this technology is obviously just incredible to see these people have this independence again. I like to play this video. This is sort of something that's recently come out, and this sort of goes one step further when you have a device here. What's happening here is they are recording his brain as it responds to the muscles he's trying to move to talk. So when you speak, you make different sounds, and they're produced by your throat and the muscles around your mouth. This brain recording device is capturing that and then translating those through a giant fridge sort of computer into this text that he can sort of directly converse with. So, I mean, this is sort of came out earlier this year or maybe late last year. So this is sort of really the cutting edge of this technology at the moment. Someone who can do thought to speech, direct words just by thinking about it, and in response to particular questions that he's been asked in this scenario. But you can imagine the real world possibilities of something like this, where someone who is unable to move or unable to talk can now have sentences in a pretty fast time, really. Again, the problem with this, as with all these, is this brick that's sticking out your head. You're not allowed to go home with these. They're big infection risk, which I don't like saying because I want to be positive about these technologies because I think they're amazing, and certainly I want them to go wireless so that they don't have this, but at the moment they do, and so these patients, they need to use them in hospitals and they can't use them at home. So when we started out, we observed this and we said, we got to fix this. There was one group that has sort of done it recently in about 2016, who were using subdural grids, which is the same. You do invasive surgery and you put grid electrodes in, but they were designing it to use at home. And certainly they showed that at least this patient had a huge amount of benefit in being able to use it in their own environment. And so that's really where we started as well. So our device is an endovascular BCI, and the reason that these other devices weren't able to be used at home, apart from the brick, is because of the infection risk. The problem is you need to get to the brain, right? And so we were able to come up with a clever way of doing this. Click, 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 click. And we wanted to utilize knowledge around blood vessels to do so, right? There are lots of blood vessels in the brain. They go to all the different sort of parts. They go to the motor cortex, which all these other devices are being implanted. But it also has the benefits that because you're not doing open brain surgery, you're not removing any skull, it's a day procedure, right? People go in for strokes. They have devices pushed up. They remove the clot. They remove cure the stroke, if you will. And the person goes home straight away. There's no need for them to stay overnight in hospital because there's nothing that is sticking out of the body. There's nothing that is warrants continued oversight on the patients when they're there. And so we wanted to take advantage of this, and so we came up with what's called the Stentrode. It's a device that can be implanted through blood vessels to the motor cortex, the part of the brain responsible for movement, and allow people to think about controlling things in their own home. And here's a little video that's a little bit old that sort of describes this. So a small puncture in the neck, it's plumbing, right? You just push a wire all the way up through to where you need to go in the motor cortex. When you're there, I made this little self-expanding device that comes out of a small catheter and expands so that you don't block any of the blood flow, and the electrodes are still secured to the vessel and to the quite close to the brain region of interest. When the person thinks about doing something, electrical activity from the brain is generated. These sensors pick up that activity and transmit it back down the lead to a pacemaker style box in the chest. And the reason this is important is because from there we can seal the patient up completely. There's nothing hanging out, there's no wires or bricks or electrodes or anything that's coming out of the patient. It's all secured, which means that they can go home arguably the same day. We obviously keep them in for a little bit because we haven't had that many patients, but there's no real reason that they couldn't go home the day of these sort of surgeries. And similar to the other ones, we connect them to technology that they can use, and away we go. The technology is scalable, which I like putting this slide in just to, because there are always questions about competitors who are making robots to do similar sort of things. But here, the infrastructure exists. The clinicians already exist. They already know the procedure. We haven't had to train them anything. They do this already anyway for strokes. The only difference is now it's easier because they don't have to remove the device. They just leave it in and they can go to their lunch or whatever they do. And there's lots of these all around the country, and that's continuing to increase. In Australia, we looked at this recently. This is like probably a year old, so I'm sure that there are more sites than this already, but there's a lot of places around that are very good at doing what's called angiography or interventional neuroradiology. People that can put stents in people, and it's an easy procedure, and they're very good at it. Let's go back to the early days. So I don't have some of the devices of the really early ones because they're just a bit shit, but this is sort of where it got to after a little while, where you can see here there's a stent or a self-expanding device made of nitinol that has these sensors on it, and the design was such that it can be delivered through a really small 1 millimeter catheter, so it can be compressed and put in, but then when it's in the right location, you remove the catheter, and as I said, it expands and puts the electrodes against the blood vessel wall. In the early days, we did have it coming out, mainly so we could do a lot of experimentation and testing on it, and we didn't have to worry about whether the wireless was or wasn't working, but certainly that's something that went pretty quickly. We started doing this on sheep in the Florey, and the Florey is an amazing place to do any early research, absolutely. They and the university were very good to us in the early days, and Clive May, who used to be there, certainly taught us a lot about neuroscience and sheep and vascular biology and all sorts of things. And so here you can see on the left there's a human brain. The vessels are obviously in blue, and the red bit is the motor cortex, or where we're trying to target. One of the beautiful things about sheep is that they're pretty similar in size. The vessels are about the same. They take the similar sort of turns, and we can get to the motor cortex in the same sort of way that we would in people. So it's a good sort of surrogate model. Here's a video of me putting one of these in. It's in a sheep, don't worry, I'm not allowed to do it in humans. And you can see that from down the bottom of the screen there on this X-ray, I've preloaded the devices. So you have a small wire that's inside a small tube, that's inside a bigger tube, that's inside a bigger tube. And really, all you do is you just push one after the other, and you just keep pushing until you get to where you need to go. Anyone who's a surgeon in the room will probably have concerns about my method, Apparently I don't use enough contrast to know where I'm going, but I know where I'm going. You just push it and you get there. But what you're meant to do is do a contrast run, right? And that's where you put this dye through the vessels and you see where the vessels are and where you need to go. And obviously there's a bit at the top that we understood was where the motor cortex is, and you can get different landmarks for that. But once you know where you need to go, for us it's as simple as, okay, we'll push it a little bit further to there, then remove everything out of the smaller tube, and then just push the Stentrode through the tube, and out it comes. And it really doesn't take very long at all. Then yeah, it's there already. That was obviously in fast time. It took about, I think this one was like 42 minutes from start to finish, which I used to think was good until I saw some of the clinicians do it in like 7 and 14. Now I don't say that anymore. But I used to be impressed with myself. But it's there, and it's pretty easy. And if I can do it in a really small vessel, then you can appreciate that a trained physician can do it in a much larger one, which they can. And so this came out about 2016 as well, when we published the Nature paper. And I think, I like putting this up here. Yeah, that's it. I like putting it up there. I'm not going to go through this, but certainly in that paper we had a whole lot of tasks that we wanted to achieve. Can we get there? Can we get there safely? Can we record when we're there? Can we do it for a long time? And we checked off all of these. The one thing we couldn't do is we couldn't get sheep to control artificial hands. They don't really have them or are very good at them. But we tried, and that was fun. So let me get to some of the clinical stuff now. So this is what the device looks like now that it's made by companies that are designed to make medical devices in approved facilities, rather than just me whipping something up in the lab at Royal Melbourne. It's now got obviously the telemetry unit that's connected to it, and all this is implanted. FDA testing. I had this slide here because I think it's important to note that the FDA testing is as exciting as this slide, but it's also extremely important. There are tests, as I was mentioning to George, where you have to get a wire and you have to bend it 1 million times and show that it's safe. You start bending, you get to like 40 million, 50 million, you get bored, and so you have to stop. But there are tests there that are absolutely critical to do, but there's no excitement in them. And I think that's where the company and the research starts the scales start tipping a bit. You need a lot of money to do these tests because you have to do a lot of them. But no sort of NHMRC or ARC conventionally, at least when we were around, is going to give you money to just bend a wire, particularly if you're trying to feed like a large team that we had back then. So I think this is where it really starts being important to have that the corporate structure that can help you find investors or other people who are willing to support the endeavour as it gets to this stage. And then the fun stuff happens. So in 2019, we were approved to do a first-in-human trial in Australia, which was Switch 1. We chose to do people with motor neurone disease, so those that had functioning brains, but they didn't have or were losing the ability to communicate and walk and so forth. I'll go through these quickly. It's a pretty easy procedure. There's pre-screening that we do where we essentially have to look to see if the brain is still functioning, which we're starting to learn is not as important as we perhaps once thought it was, and that they still have vessels of the right size. Most people have one big vessel and one small one. That's just how it is. In the early stages, we wanted to make sure that they were pretty similar in case something went wrong. Nothing went wrong, so I think that's also something that's starting to shift on our pre-procedure assessments. We then look at the vessel and we decide where we're going to put the device by using co-registered MRIs and CTs. And then when I say we, then a trained physician will put one of these devices in. You can see on the bottom right, the bottom left is just an image of his hands, and the bottom right, you can see the device just moving up and straight through the large blood vessel to the motor cortex. when we're there, they just pull back on the catheter and it self-expands, and it's where it needs to be. There's a pocket that's in the pectoral region, and then you can see afterwards on the brain scan as it's sort of rotating around. What's good is if you probably can't see anything, because that's the point. We don't want to have all these wires and all this stuff hanging out. And certainly, this is what the vessels look like, and it would look the same even if there wasn't a device in there, although you can sort of see where the lead is coming through. All the safety endpoints from Switch 1 were met, which is obviously very exciting. We've just clocked over maybe 12 years of cumulative use with these devices without any serious device-related adverse event. Unfortunately, of course, a couple of the participants with motor neurone disease are no longer with us, but there are a number of them that still are, and they're still using the technology years after being implanted. And certainly, this was from the Australian trial. We've branched out into the US, and a couple of weeks ago we completed the implantation of our sixth patient to round out the early feasibility study, the study you have to do before you do the next study to prove that it's safe before you can do the selling and get it out to the people that need it. So a long journey, and it's as it should be, I suppose. No device migration, doesn't move, no. Is in the right spot, yep. How are we going for time? Because I want to do some videos. You good? Great. So this is Graham. He's the first gentleman that was implanted, the first astronaut, if you will. And what he can do here is use the device in conjunction with eye tracking, not only to make a click, but also we were able to give him the ability to do like a click and zoom, so he can zoom in on particular parts of the screen or particular letters, and here he's typing out a message. He actually wrote a book using this system, Technopathy for Beginners, and hopefully that hits the bookshelves soon. I think it's going under the edits, but no, he was a lovely man, and certainly we'd be indebted to him the whole time. Yeah, let's get to videos and then we can have some fun questions, right? So here, this is one of the patients that's in the US, and one of the things that is important for them is he's obviously a lot more disabled than Graham was. This guy, and the way he's intubated, he's in hospital, he's completely locked in, he can barely move his eyes. Eye tracking's not really an option because they can't focus it properly. But with this system that we've been able to hook him up to an iPhone and an app, he's able to use it to control a reporting message that can be sent to a clinician. And for them, that's really important. How can this person tell other people what he needs if he needs to be turned because he's sore on one side, if he's hungry, if he's needs to go to the bathroom, whatever it is. I can't imagine what it's like not to be able to talk, as I'm guessing you have probably figured out by now, but certainly these guys, giving them back a voice or some way to communicate is really, really important, and I think all the groups around the world who are looking into this problem, I'm really impressed with all their work. I'll show you this one. This is Phil. He's one of the other Aussie fellas, and this is the first time he's used the technology.
Just any old file. Try and stare at it and think about moving for a long time, and it'll start zooming in. Yeah, and release. So you made a click, your first click with your brain. What? Okay. So I want you to click on that by staring at it and thinking about moving or planning the movement. Yeah, and then try and relax back. Right, so you open up Word.
Holy moly.
Now, I want you to open up exactly the same thing. Open up a blank document for me. So now just start typing whatever you want to type. What are you looking to type?
W-O-W.
Very, very close. There you go.
Wow.
It's not quite wow, it's Wooloo.
Well, you was trying to say welcome.
That's wow.
How was that?
That's my first attempt at trying to do that.
Yeah, it's amazing.
Yeah, so we've been very lucky with the patients that we've had. This gentleman, Rodney, he also a champion like the others, and certainly one thing that they've found really, really useful is that they can now connect on WhatsApp, and they can sit there and they can text each other, and they can talk about how the system's working, or how their disease is progressing, or how they're feeling, and they can have a sense of community again that they perhaps felt that they had lost before they got their prognosis. So this is these two guys having a talk about Tom before he gets on stage, and just having a nice chat, and it is really nice. I went round to his house probably about a month ago, where we were. This is when the Tigers still had a chance of making the finals, and we were able to sit there and just have a chat about, like, I'd be texting to him and he'd be able to read it and he'd be able to text back, and it was just really nice. And I think some of the things that these guys miss more than anything else is just being normal and just being able to talk about things that aren't their condition for a change. And certainly they've loved it, and they're asking us to do a whole lot of other stuff. So we're getting them to, we're teaching them that they can play Pong, and we're teaching them how to use the system without eye tracking. So we want to make sure that they get the best outcomes, whatever their condition is. And there are some people who can't use eye tracking, and for them we need to have a way that they can just use the Stentrode and that system to navigate around a screen, or to type or to text or whatever it is. There are others that can use eye tracking, and if you can and it's working well, great. Let's use that as well. Like, so we really want to make sure that whatever the person can or can't do, we can help augment that and make them the best that they can be. Including, as in Phil's case, turning on the TV in the middle of the night just to freak your kids out. So he liked doing that. So that's the system now, right? You get the motor intent. We turn that into, transmit that, and interpret it, and then use that to control something. And the thing that we're really excited about at the moment is we don't know what technology is coming in the future. We know what's here now, and we know how to try and connect to it. But as more things start coming out, as smart homes get better, there's more stuff people want to connect to. It started off obviously with computers, and they do spelling. Now they can connect to the TVs. Now they can connect to their lights. They can play Battleship. They can now play with people who aren't in the same room as them, just online. There's a huge amount of things that are coming out for these guys, and I think it's going to be really exciting to see what they can do in the future, whether it's lights, glasses, computers, who knows. And so I think I'm probably up for time, but this is the last video just of Rodney using this system to turn on a light. And again, this was his first try. We're starting to develop these boards and starting to try and understand what people want to do, which is obviously often quite different to what engineers think people want to control. And so for him, he's like, I'd love to control my lights. And we're like, all right, that's a lot easier than getting you to control some exoskeleton that we would love to build. But that's what you want, so you got it. You can see him here just using this pretty simple program. Click on what you want it to do, and then the light goes on. And stuff like this can make a really big difference to these people. Trials completed. So here's the team. There are a couple of fellas sitting around there that might recognise themselves. Tony and David, who have been with us right from the very start and seen us grow and had a lot of patience watching us do so, no doubt. And so I'd love to obviously thank all those team, and then the bigger team as we've started to expand into the US. We're now about 70 people now, so this isn't all of them obviously, but like you were saying, Mark, this is a huge multidisciplinary collaboratory organisation. We have all sorts of people from all sorts of different professions, whether it's engineers, scientists, clinicians, researchers, marketers, lawyers, all that sort of stuff now. So I think it's really fascinating, and it's really been exciting to work with such an amazing and diverse team, and we're having a lot of fun doing it. And I know it seems like we've come a long way. It feels like we've got a lot further to go, but I think we're making the right track. And so with that, I'll probably leave it, and I'm happy to take any of your questions, regardless of what they are. So please feel free. Thank you.
So, thank you, Nick. Now we have, I believe, a couple of roving microphones. So I'll indicate who will take the questions. I warmly invite you, if you're comfortable, to share your name, introduce yourself for the room, and for those who are joining us online. That's why the microphones are so important, so those online can hear your questions. And I'll invite Nick to step up and to answer the questions. I have a very eager question here in the front. Thank you.
Hi, Eric Peterson. I've been playing around with tech for decades. Saddened to see you showed an example of someone using a QWERTY keyboard when they were looking at things. QWERTY was designed 2 centuries ago to slow down people typing fast, to stop the keys getting in the way of each other. So all the frequent keys are actually way out at the sides. Dvorak came up 120 years ago, nobody knows about it. Palm Pilot, and I think a lot of the handheld computers did a lot of work with far superior keyboards, which presumably would minimise the eye strain. Have you looked at any of that yet?
Yeah, we made our own keyboards as well. We've got some that make it easier to do less sort of clicks, particularly for those who don't have the eye tracking. We're like, well, how can we give you the most amount of benefit with the least amount of effort? And so, yeah, you're absolutely right. That keyboard was one of the eye tracking programs that they use it. I agree with you that there are better ways to do it, and there are. And I think certainly what's going to be exciting in the future is what's going to happen with the predictive text. That already increases speed a lot. And when you go a step further, and instead of just predictive text, you get the ChatGPT style answers that can give you full sentence answers with just a single click, then that's where speed's really going to ramp up for some of these people. Thank you.
Thank you. We'll take a question here. Yep, here in the front. Yeah. Thanks, George.
Hi, George Dimitroulis. I'm the founder of Maxonic here. Do you see an application for this in normal people? In other words, to be able to command things without necessarily verbalizing anything? The lazy people like me who sit back and change channels without having to press the remote.
There's always someone like you in the crowd, yeah. So look, it's possible, right? If your brain's working, then you'll be able to control things. I think for us in the company, we're certainly focused on those who can't do it themselves and how we can get them to a different level. But I mean, I think you see a lot of industries like the plastics industry, right? It used to be for burns victims or people who are missing limbs and things, and now I think the biggest market is just people who want to look different or prettier or whatever it is. So I think there's a lot of space for this to turn into a consumer product, but certainly that's not where we're heading. But I hear what you're saying. No, you can't have one.
Thank you. We have a question on my right.
Hi, my name is Alon. I'm a STEM educator, but before I got into education, I did my honors in neuroscience. And I'm just curious as to what exactly you're recording, because you're not inside neurons recording action potentials, like with patch clamping, but you're also not doing whole brain waves with, as you said, a toy. You're somewhere in the middle. What are you recording, and how are you then classifying that into left, right, up, down, in, out, or however many different classes exist for the responses?
Yeah, so what we're recording is local field potentials, which is about 1,000 neurons or so, and they're all talking together when you're trying to do something. So in the brain, there's a general hum of activity, and little neurons are doing different things, but when there's a big command like squeeze your fist, for example, there's a lot of these neurons that bunch up and all sing the same song, and that's where we grab them, the local field potentials, and that's where we can interpret these and say, oh, all of these neurons in this area said this thing, so let's do that command, or all of these ones over here said something, so let's do this command. That's about as high level as I can put it, but yeah, local field potentials.
Thank you. Yes, question.
Hi, I don't know if that's on. I'm Marie Sierra, and I'm the Dean of the Faculty of Fine Arts and Music, so I'm going to ask you a question that an artist would.
Good.
If I were locked in, I'd want to be able to draw. And I have a brother-in-law who in the UK who's an illustrator, and he does everything on a tablet, absolutely everything. I have composers in my faculty who do everything electronically. Have you thought about making that jump to being able to create? Because we know that there's a relationship between creativity and well-being. Music therapy has proved that very strongly, and many other disciplines have. Is that a next stage rather than just being able to type?
Yeah, I mean, I think that's an amazing suggestion, and certainly we're at the point now, fortunately, where we can say, okay, well, there are some certain things we need to do to prove it works for regulatory reasons, but there's certainly a lot of other stuff that people want to control, and that's a great one. I mean, if, like you're saying, if anything that can be done on a computer, these men and women would be able to do. So that's great, and I think if there's a way that we can work with those program guys and make the programs more accessible or easier for whatever limitations that the people have to use, and then yeah, that's a great suggestion. Thank you.
Thank you. Yes, a question here with Jane. Thank you.
Thanks, Nick. So it's operating on the motor cortex, and I'm just wondering, how far away or how much distance does it get? Does it pick up anything from the sensory cortex, or have you noticed any emotional sort of things happening with people when they're using this apparatus? Or it's very local?
Yeah, no, that's a great question. I think we're really focused at the moment on getting a response from an intentional movement. So, and you see the guys at the start, we were really lucky that they initially had some movement, so we could see, or they could tell us, I'm trying to do something now, and we can collect it and know what we're looking at. I think that there's a huge amount of data that we're collecting that that would be able to answer some of the questions on are we picking up things that are not motor cortex? Are there better ways that we can do that? And I'd love to have another 1,000 computer scientists working on these to try and figure this out. But certainly for us at the moment, we're really focused on just how can we do that. And I think as we've also only had a relatively small number of patients, it's hard to discern what is normal for someone with this condition and what their mental health is like on a normal scale compared to what it is with this. But perhaps there's some markers in there that can help tell us that we'll be able to look at in the future. But that's a great point. Thank you.
Thank you. Yes, question here in the back.
Thank you very much for your presentation. My name's Cosmos, and a layperson. I was just wondering, what would be the impact if you place this, the stent in a different part of the brain, such as the frontal lobe? And, what sort of impact would you have, given that the frontal lobe, has something to do with executive functions, control, personality, and such, and whether it's, for example, whether there's a possibility for mind reading?
I think certainly there's a lot of work that's been done by Medtronics and those folks looking at other neurological conditions like epilepsy and PTSD and depression and that. And there's certainly vessels that go deep into the brain where our device could target. And so I think certainly the next step would be how can we solve other neurological conditions and how can we potentially make those technologies that exist even safer. But you're right, the brain's a big place. There's a lot going on. There's a lot that can be done there. And so I think there is a way that you could extract information, but don't be fooled. The information is hard to interpret. There's a lot going on. There's a lot of noise. I don't think it's as simple as watching a TV screen of your brain. It's really requires a lot of interpretation and translation of these ones and zeros that we're getting out to reconstruct it into something that might be useful. Maybe ask some of the Silicon Valley guys what they're thinking of that, but we won't be going in that space anytime soon.
Is your follow-up question related? Thank you.
Yeah, the follow-up question actually relates to the fact that you're using action potentials. We also know that the brain works on neurotransmitters, chemicals. Is that an area that you're also focusing on, and I guess the interface between the electrical as well as the biochemical?
Yeah, great question. I mean, I think for us, and one of the reasons that we're quite different from a lot of the other technologies that are out there, but perhaps also why we've been able to leapfrog all of these technologies that have been out for decades, is we really went from the other side, and instead of saying how much can we put in and how much can we collect, we sort of took the approach, even if we only had one sensor measuring just a handful of neurons, what can we give this patient, and then ramp up from there. And so for us, we know there's a huge amount of information that neurotransmitters and what have you that when could record but perhaps not, but really we want to make it as simple as possible, as reliable as possible, something that the patient can use after they've been implanted as soon as they wake up. And for us, that was, let's just see the biggest signal we can and give it to you, and away you go. And if you've got time to learn more, then we can teach you more. So I think there's certainly scope for that, but I think we took the right approach by making it as simple as we could, and we'll sort of keep going from there.
Thank you. Just see, we've got some questions here. Adrian, I'll take your question, and then we've got two more questions here, and then we might be close to wrap up.
Thanks, Georgia. Yeah, great, Nick. Adrian Pearce from the School of Computing Information Systems. I'm thinking about signals going the other way. I mean, obviously the communication's kind of half duplex at the moment. Probably a naive question from my perspective. I have seen a little bit of research a long time ago, and I'm not sure, not necessarily, getting information into the brain, but possibly just tweaking control, and whether, there's things you can do that makes the interface easier to use maybe, or obviously maybe in epilepsy or something you can do it. Obviously you've thought about it. What's the thinking there?
Yeah, we have thought about it. Obviously epilepsy is a great one. There are a lot of conditions where putting stimulation back in will impact the patient in a positive way. So Parkinson's tremor, that can stop if you stimulate. Epilepsy seizures can be stopped if you stimulate there. I don't really understand how depression works, but I know they're stimulating for that as well. And so I think there is scope to start putting information back in. I know that when we started, some of the folks at DARPA made these incredible robotic arms that they were connecting, and one of the problems they had with them is the patient didn't know when they were grabbing things. And so for them, how could they get some feedback so that the patient would know, like, oh, okay, I'm now grasping this, I'm grasping it just the right amount, not to drop it or not to break it. And so I think, certainly for the neurotech space, the sort of the feedback is going to be one of the first things that should be looked at.
Cool, good response.
Thank you. Saw that there was, yes, a question here. Thank you.
Thank you. Carl Xing, medical student and part of a student project at the Melbourne Bioinnovation Student Initiative, where we work on the EEG-controlled brain-computer interface. So very excited for your talk. My question's got to do with sort of the development journey of the Stentrode, and I'm wondering if you could share what are some of the main challenges and uncertainties that you faced in developing this project, especially in the early stage, starting from the conception of it.
How much time we got? So I think
just the key ones.
Yeah, just the key. Okay. I think the first thing that was really important, that wasn't hard, but I think is just really important, is to make sure that you have a really good team, because there are going to be things that are really hard, whether it's technologically or fundraising, or just the science isn't working, or things aren't working. And if you have a really good team around you, then it's all going to be fun and you're going to have a great time. So I think that that's really important, and certainly David and Tony are the guys that knew a lot and helped us sort of put a team together and helped us understand the science that we didn't know at the time. So I think having a good team is certainly number one. In terms of the things that were tricky, I suppose how to make a piece of technology that has two states, one that's really small and tiny to be able to be pushed through the tube, but then can also expand when it's in the desired position. But not only that, but how can we do that with all these sensors on it, and also in a way that's biocompatible, but also gets incorporated into the blood vessel. And there are all these and it has to keep doing this. And so that was challenging, but also probably the most fun bit. So.
Thank you. And yes, a question down the front. Thank you.
Hi, thanks for the presentation. My name is Ben, calling Benny. I'm a current biomedical engineering student, and it is impressive technology about like how to put the stuff into the brain. But I'm a bit curious about like if the patient shift their mind, they want to take it out. Have your team consider about like how to do that, and are we available to do it, and what's the risk?
Yeah, it's a good question. Removal. So we did a study recently showing that at least out to 7 days it could be removed. The general principle though is, and it's the same with cardiac stenting, you put one in and it just stays there and it's safe and it's biocompatible, so nothing happens. If in these patients you put devices in because their vessels close. If you put a device in to open the vessel and the vessel closes again, they don't get rid of the first one, they just put another one in. And so we're taking a lot of advice from these guys who have been doing this for the stenting work for decades, and their opinion is once it's in, leave it in. If we absolutely have to get it out, sure, there are ways to do that, but most of the people we've spoken with have never taken one out because it's safer to leave it in than remove it. And so I think for these patients as well, the difference is that it only works if they want to use it. So if they don't want to use it, it's not doing anything. It's not getting information out if they don't connect to anything. It's not putting anything back in. So it's passive and it's sort of off all of the time unless they turn it on, which sort of makes it safer and in their control on when it's active and used.
Thank you. And that, I'm afraid, is our last question. And with that, please join me in thanking Nick and his journey. Thanks to our speakers tonight, Nick, and to Mark Hargraves, and to you, the audience, for coming along tonight and for your absolutely fantastic questions. If you've missed any of our events this week, or you'd like to see this one again, or indeed share it with others, all of the Innovation Week recordings will be available online soon, and you'll find them through the Melbourne Connect website. And if you're not already on our mailing lists and you'd like to learn more about what's happening here and how you can get involved, that same Melbourne Connect website is a fantastic place to sign up, find out all of the incredible events that are coming, and learn more of what our incredible partners and what we, the university, are doing with them. Our next major event is the future of smart surfaces. And if you thought this was seeing a window into the future, that one too will really blow your mind. And it's being held here again in November. Thank you for joining us. Thanks for a really warm conversation, and good night.